Good afternoon and welcome to the webinar. This is Caterina Cremonesi, Sales Manager at Prodotti Gianni. Today we have the pleasure to host Ignazio Mazon, who is a Strategic Account Executive at Arima Genomics. Arima Genomics is distributed in Italy by Prodotti Gianni. Ignazio will give us a presentation entitled Understanding the Genetic Basis of Cancers with 3D Genomics. If you have any questions, feel free to write them down in the chat. The webinar will be recorded and you can find it in our YouTube channel. That's all. So thank you for attending the webinar and enjoy the presentation.
Thanks, Caterina. Thanks so much for the kind introduction and good afternoon, everyone. So I'm very happy to be here today to talk about 3D genomics and also have the opportunity to discuss with you how important it is to study the 3D genome structure to understand the genetic basis of cancer. So I hope also that after this webinar, you will get a better understanding on how you could use 3D genomic tools to support your research projects. My presentation will be divided in three parts. The first one is an introduction on what 3D genomics means, and also I will talk a little bit about the high CSA that is offered by Arima Genomics to study 3D genome conformation. I would also provide you in a second part with a few examples on how to apply 3D genomics in cancer research. And finally, I will give you a short and brief overview about the product and services that we are offering at Arima Genomics, and we are distributing by Prodotti Gianni in Italy to also study 3D genome conformation. Arima Genomics is a leader company in the 3D genome space, and our goal is basically to provide solutions to academic and also clinical researchers to understand the 3D genome structure, and ultimately to help human health and also to drive discovery. We are a company that is based in San Diego, in a very nice city called Carlsbad. This is northern from San Diego, close to the beach. And then we're about 35 employees right now in the company and growing. We have 5 products right now in the market. And then we have about 700 individual labs that are already using our technology. And I think about 500, a little bit slightly over 500 publications citing the use of our products. As Caterina mentioned, our products are distributed in Italy through Prodotti Gianni, who is our exclusive distributor. First question you will ask myself is why pursuing 3D genomics? So basically we know that today we know that 3D genome structure or chromatin conformation has a big impact in cellular function and more specifically on gene regulation, DNA replication, or DNA repair. While most of the NGS approaches study the DNA in a linear way, we all know that the DNA is folded in the nucleus and form different 3D genome structures. So basically 3D genomics study not only the DNA sequence, but also the DNA structure and the impact on cellular function. So as you know, the DNA is organized in the nucleus in different ways. So the highest level of organization of the DNA is the chromosome. And the chromosomes, as you also know, they are located in the nucleus and in different territories. So in generally, in healthy cells, chromosomes are not interacting to each other, with each other, but basically they are remaining in their specific territory. However, in tumor samples, genomic rearrangements between different chromosomes are very typical and in many cases are also related to tumor progression. So this is a feature that is happening in tumor samples. Within the chromosomes, you can also find what we call A and B compartments, which reflect basically open chromatin and closed chromatin. And within those A-B compartments, you can also find other type of 3D genome structures, like topological associated domains, which are self-interacting domains, or also chromatin loops, which normally are formed when a promoter enhancer are interacting. So what Arima does is to study these 3D genome structures, because we know that these 3D genome structures harbor biological information, and it has also been shown that this 3D genome organization has sometimes pathological implications. Before I'm explaining you what is the technology that Arima Genomics is offering, I would like to stop here and just highlight that 3D genomics is a critical part of any multi-omic approach. As many of the researchers are doing today in their research, people are combining different omic technologies, just for example, to study gene regulation. So 3D genomics is becoming also a very important part of this puzzle, and is really linking genomics and also epigenomics by determining how chromatin conformation change gene regulation. So what is special, or what is Arima Genomics offering you to study the 3D genome structure? So basically, we are offering a powerful and very sensitive Hi-C approach, which is basically an assay that is called high throughput chromatin conformation capture, and will allow you to study how spatial and temporal changes in chromatin conformation alter gene regulation and also cellular function. So basically, Hi-C is a combination of a proximity ligation assay and also an NGS approach. And what the information that it will deliver is a genome-wide map of chromatin interactions, which will allow you basically to study the spatial organization of the genome and how this is related to cellular function. So how does it work, Hi-C? So it's very similar to other conventional NGS approaches. Here in the upper part of the diagram, I'm just showing a conventional NGS workflow. In this case, is a DNA sequencing workflow. So as you know, the first step in any DNA sequencing approach is just to start with your sample to extract and purify your DNA. Then you will share it with a sonication device. Then you will prepare your library, and then you will sequence. You know, with DNA sequencing, you can, for example, look and compare sequences from different patient samples and look for mutations. With Hi-C, the workflow is very similar, but the only thing that is the main difference is basically the sample prep. And similar to other chromatin-based approaches like ChIP-seq and ATAC-seq, the first step of the Hi-C is basically cross-linking the sample. In Hi-C, you can start performing your experiments with cell lines, primary cell lines. You can also use frozen tissue samples. You can also use FFP samples. And the first step, as I mentioned, is just to cross-link with an agent that normally is formaldehyde. We normally use and recommend at Arima to use 2% of formaldehyde. Second step is just that once you have stabilized the chromatin interactions with the cross-linking agent, then we digest the sample with a cocktail of restriction enzymes that these are provided by Arima in the Arima Hi-C kit, and which is also patented by Arima. And then what you will generate are chromatin fragments that you will mark with biotin, and after that you will proceed with a proximity ligation assay in the way that the fragmented chromatin pieces are ligated together, and what you are generating here are chimeric fragments, which are represented genomic regions that are in proximity in the nucleus space. Okay, so those fragments that are in close proximity will ligate with a higher frequency. Okay, and these chimeric fragments will then represent regions that are in close proximity, meaning interacting. Okay. So once you have these fragments, you will once you have generated these chimeric fragments, you will purify the DNA, and then you will pull this DNA with streptavidin bits because it's biotinylated DNA, and these biotinylated chimeric fragments are the ones that contains the 3D genome structure information. Okay, so you will prepare a library using one of our library prep kits that we develop at Arima, and then you will just sequence with per N sequencing. Once you have your sequence chimeric fragments, you will map them to a reference of genome, and that way you will be able to locate those areas in the genome that are, or identify those areas in the genome that are interacting. So basically, we normally use open source computational tools to analyze the data. And basically, what you are generating is a genome-wide interaction map, which are called contact matrix. Okay, and these contact matrices will represent the frequency of interactions between different genomic regions. So why you should use Arima Hi-C technology, or what is special about our technology? Is basically we develop an assay that has been overcoming some of the main challenges that are presented by traditional and conventional homebrew Hi-C methods. The first one is the lack of versatility and the need of millions of cells just for the experiment. With Arima Hi-C kit, we are able to perform Hi-C experiments with as little as 50,000 cells. Why is this relevant? Because we are opening the door to perform Hi-C on clinical samples when using our technology. Another challenge that is also overcome when using Arima Hi-C technology is the time for the experiment. Traditional homebrew Hi-C methods are time-consuming, requires at least 3-4 days just to complete all the process. But with Arima Hi-C technology, you will just need only 1 to 2 days to complete the whole experiment. Just to give you an example, the Hi-C itself, it's done all the reaction in one tube in just only 8 steps. So it's very simple. There is no loss of material. And then after that, you will just only need to prepare the library. Something that we have also seen that is very good with the Arima technology is that when comparing our assay with the conventional Hi-C homebrew methods, we have seen that there is a better quality, but which is represented by the higher percentage of long-range SINS interaction that you will obtain with the Arima Hi-C technology. This is the QC metric, and this is basically the interaction that you will be looking for just to analyze the 3D genome structures. In addition to that, there is also a reduction in the sequencing cost, which was also one of the main challenges of a conventional Hi-C protocols, which normally require billions of reads just to perform to complete the experiment. With the Arima Hi-C technology, you will just only need 300 to 600 million parent reads just to perform high-resolution Hi-C experiments, which means just to detect promoter-enhancer interactions. Okay, you will tell me, okay, that's still quite a lot of reads in comparing to other omics technologies, but it's still a significant reduction of the number of reads that you will need to sequence in comparison to other Hi-C methods. And last but not least, also quite important that is the fact that the Arima Genomics Hi-C protocol contain built-in QC steps, which allows you also to track the quality of your samples at different stages during the protocol. And that's also very important because at the end of the day, as I mentioned, you will need to sequence quite deeply to get your results. So it's nice to get into to send your samples for sequencing, knowing that the quality of your samples is good, and then you are securing good results before sequencing. So what type of applications or how you can use basically the Arima Hi-C assay? So you can use it for different type of applications. I already spoke about how important it is to study 3D genome structures. So you can really use Hi-C to identify new topological associated domains, to do differential analysis in terms of chromatin loops between, for example, a healthy sample, a tumor sample of a sample that has been treated versus non-treated. But also the Hi-C data contain information about the promoter-enhancer interactions. So it's also a very helpful tool in combination with other multi-omic approaches, just to study gene regulation or either other cellular processes like DNA repair or DNA replication. So with the promoter enhancer interactome, you can also get some insights on gene regulation. In addition to that, Hi-C will link epigenetic with physical chromatin interactions. And in fact, we are also offering a single cell 3C solution that will allow you also to profile in just a single cell both the DNA methylation profile and also the 3D genome structure. And last but not least, which I think might be also very interesting for the audience at this webinar, is the fact that the Hi-C data also contain information about genomic rearrangements that are happening in the genome. So with the different bioinformatic pipeline that you will use for example, promoter-enhancer interactions, you could also use the same set of Hi-C data to detect structural variants in your genome of interest. But before providing you some examples on how 3D genomics and the Arima Hi-C technology can be applied to your cancer research, I would like to stop here and talk a little bit about Hi-C data visualization. I mentioned before that with Hi-C data, you can generate genome-wide interaction maps, which we call them contact matrices. Okay, these contact matrices represent, again, the frequency of interactions between different genomic regions, and they can be visualized by using different bioinformatic open source tools existing out there, and by using what we call heat maps. So here on the left side, you can see a heat map that has been generated by the telomere to telomere consortium to scaffold a complete human genome sequence. What you are seeing here is basically the chromosomes which are lined up across the top and the side. Okay, and each point of this heat map represents a physical interaction between two genomic regions. If you look at the dark red points, which is on the diagonal of this heat map, you can observe that most of the interactions are happening within the same chromosomes. Intrachromosomal high signal is what you are observing here in this heat map. This is a very characteristic in healthy cell lines where, as I explained before, chromosomes are located in different territories in the nucleus, so they are not interacting within each other. Okay. Let's have a look now to a comparison of the differences between a normal karyotype, represented here by this heat map from a human cell line, versus an abnormal karyotype represented by this heat map, which is corresponding to a tumor cell line. On the left side, you could see again that all the interactions are happening primarily intrachromosomal. So meaning that, as you can see, that all these red spots across this diagonal. However, if you look at the red figure, you could also observe that there are a lot of red spots which are off the red diagonal, which are representing interactions between different chromosomes. Okay, in that case, these are interchromosomal translocations, what we are showing in this picture. So what is great about these heat maps is that you do not only use them to detect rearrangements in the genomes, but you can also look for interactions that are happening within the same chromosomes. You can really zoom in and out to look for interactions at different resolutions. This is an example of about, let me see, it's about 800 kilobases region. And what we're showing here is the different interactions that are happening in this region. And here I'm showing with different colors interacting domains, which are representing regions that are interacting. So let me find the laser pointer, sorry. As you can see here, these red dots are representing reads, representing an interaction between the yellow interacting domain and also the green interacting domain. You can also see another a strong interaction between the blue or violet interacting domain and also the black domain, and also a very weak interaction between green and blue. If you make a model out of this observation, you could imagine that, you know, yellow and green are really very in close proximity in the 3D genome space, and then there is a loop that has been generated by these two domains that are interacting. So this is the first part of my presentation. I don't know if you have any questions, but please feel free to add any comments on the chat box. So let's now talk about different situations in which 3D genomics has been applied to study cancer research. The first case I would like to show you today is a case in which researchers have used 3D genomics and specifically Hi-C to understand cancer disease mechanism. In this very exciting paper that has been published, I think, in Nature Genetics by Clockton and colleagues, what they are showing is that by integrating Hi-C RNA sequencing and also ChIP-seq data with CTCF, they have observed that there are differences in the 3D genome structure between healthy individuals and also TALL patients, leukemia patients. And then what they also demonstrate in this paper is that by doing Hi-C, they were able to identify that fusion that was enabling direct interactions between MYC oncogene and also an enhancer region that are far from this oncogene promoter region. So what I'm showing here in this slide is in the upper diagram. You can see here the linear conformation of an oncogene in comparison to its enhancer or potential enhancer, which is 2 megabases away from the oncogene promoter region. Here on the right, when these researchers perform a Hi-C, they could observe that both the oncogene and the enhancer regions were part of two different 3D genome structures. In this case, two different topological associated domains, as it is also represented here on the right side in this figure. Okay, here you see that both the oncogene and enhancer are part of two different topological associated domains, and therefore they are not interacting to each other. What they observed is that when they were performing Hi-C on TALL patients, they observed that by Hi-C, both the oncogene and also the enhancer region, in the presence of a transcription factor binding, both regions were now part of the same 3D genome structure, in this case, the same topological associated domain. So under the presence of this transcription factor, both regions, the promoter of the MYC oncogene and the enhancer regions, were part of the same topological associated domains, which was allowing this enhancer to bind to the promoter of the MYC oncogene and then promote its expression. Moreover, the researchers in this publication, they use a drug or pharmaceuticals just to downregulate the expression of this transcription factor to avoid the binding on the enhancer region. And what they observed by using 3D genomics and specifically Hi-C technology from Arima Genomics is that the original two topological associated domains conformations was restored after treating these patients with this drug, and that way they were able to explain the mechanism by which this drug was helping these patients with survival or increasing survival in these patients. So, which are the takeaways of this case is that the 3D DNA folding and Hi-C help researchers to understand how MYC is regulated in a healthy state, but also how it's misregulated in patients which were suffering of leukemia, and also they also use Hi-C to understand how MYC regulation and expression is restored after treatment. This is another case in which I would like to show you, in which researchers have used 3D genomics and also Arima Hi-C technology to identify potential therapeutic targets and response in cancer. Basically, the researchers that published this paper that were part of the Carbon Institute, they did a study to understand gene expression and regulatory landscape following tumor treatment. And for that, they use a battery of multi-omic approaches. And what they did was to take patient-derived metastatic breast cancer cells and graft them into a mouse model. And the mice were treated either with a epigenetic drug called the cytarabine, which is an hypomethylated agent, or with just a saline solution. And as I mentioned, they perform different omic assays to analyze or to study the gene expression before and after treatment. Also, the DNA methylation profile in these mice before and after treatment. Also, the binding profile of a specific transcription factor by chipstick experiments. But they also perform Hi-C, genome-wide Hi-C, and also captured Hi-C or targeted Hi-C experiments to look at how the 3D genome structure of these mice were changing after the treatment with a hypomethylated agent. So here in this slide, I'm showing you some results on the genome-wide Hi-C experiments that these researchers perform in the mice that has been treated and non-treated with the cytarabine of this hypomethylated agent. And what they observed after treating these mice with a epigenetic drug was that there was a decompactation of the chromatin that has been observed thanks to the genome-wide Hi-C experiments. Moreover, they wanted also to understand what was the mechanism of this the methylating agent, the cytarabine, and how it was acting in specific genes that were related with a better prognosis of the disease that were experienced by the mice that were treated with this hypomethylated agent. What they discovered is that many of these genes that are related to survival, which were estrogen receptor genes, were gaining in enhancer active promoters, and this was basically observed thanks to performing capture Hi-C experiments. Okay, so again, I just wanted with this case to show you some more evidences on how to use a Hi-C technology and basically 3D genomics to study or to understand how important is to study 3D genome structure, not only to understand gene deregulation in breast cancer, as these researchers did in this case, but also to understand the mechanism by which a drug could suppress tumor growth. Let's talk now about the last application that I think is going to be very relevant to some of the research that you are doing at your institutes. So basically, it's the use of 3D genomics and also Hi-C to detect clinical relevant genomic rearrangements. And to provide you, with some more insights on how impactful could be to use the Arima Hi-C platform, I wanted to provide you some information about one of the cases in which, or the first cases in which Arima Genomics was involved in collaboration with some doctors, just to help diagnose the cause of cancer in a specific patient. And this case is about a girl, a pediatric cancer, a girl, a 14 years old girl that came to the NYU hospital in New York and was diagnosed with a stage 2 glioma. Okay, so initially this patient was treated with a total resection of the tumor. But what happened is that after 6 months, this patient came back to the hospital because experienced a very rapid progression of the tumor. And initially, the doctors in this hospital, they ran different tests just to detect gene fusions by DNA sequencing and RNA sequencing panels. But all these diagnostic tests failed to provide a cause of the disease, and the patient was treated with chemotherapy. However, Dr. Snoodle, which was one of these doctors involved in the diagnosis of this patient, he knew about our technology, so he got in touch with us and he sent us the sample of this patient, and then we ran a Hi-C at our facilities. So what we were able to discover was a novel gene fusion that was involving PDL1 gene in chromosome 9. And for those that are not familiar with PDL1, this is a gene that basically helps the immune system to distinguish between tumor samples and healthy samples, tumor cells and healthy cells. So this was a very exciting evidence or finding for Dr. Snoodle, and what they did in their hospital is just to take the sample of this patient, and then they ran immunohistochemistry to look at the expression of the PDL1, and they were able to confirm that the PDL1 gene was overexpressed in this patient. So what is great about this story is that, there was already an existing drug in the market that is used to diminish the expression levels of PDL1, and then the value of us collaborating with this doctor was that we were able to rescue one patient from a chemotherapy treatment to a more targeted and existing treatment that allows the patient to have a better life. And as of today, we know this patient is still stable. After being involved in a few similar cases at Arima Genomics, we thought, okay, wow, this Hi-C technology looks to be very sensitive and helpful to detect genomic rearrangements. So we start collaborating with many other hospitals in US, and then basically by doing retrospective analysis and by looking at samples that has been analyzed with other technologies existing in the market, like Archer diagnostic test for detecting RNA fusions, also samples that has been analyzed with FISH or standard karyotyping. And then we ran about 100 samples, and we were able to detect in all the samples the same gene fusions that has been detected with these other methods. Moreover, we expand our analysis to try to understand if this technology could be robust also across different type of tumor types, and then we've been able also to confirm these results. We even went deeply in our comparative analysis, and then we also took about 32 samples from NYU that has been analyzed with the Archer diagnostic technology, And again, different type of tumor samples, adenomas, breast cancer samples, head and neck, lungs cancer samples, and again, we were able to detect the same fusions that were detected with the Archer technology, but with our Arima Hi-C technology. So why is this very relevant and why we think it's a very exciting story is because we know that as of today, 54% of the advanced cancers are driver negative. So meaning that there are no NGS technologies that are able to detect the drivers of the disease. Because of that, we have also engaged in new collaborations with other hospitals in US, and so far, I think this slide is a little bit outdated, but as of today, I think we have analyzed about 50% of driver negative samples in our facilities, different type of cancer samples, pancreatic, sarcomas, also hematological cancer samples, or tumor samples, and we've been able to identify in half of these driver negative samples, clinical actionable or fusions that are involving clinically actionable genes. This is very important, as you can imagine, because this means that in half of these samples, we could have helped doctors to find a targeted treatment for those patients. After performing all these experiments, we have concluded the following: that Arima Hi-C offer several advantages over other technologies for SBS detection. The first one is that it's compatible with any type of samples, including FFPE, which this is really important because there are other technologies like RNA sequencing panels or long-read sequencing, like PacBio or Oxford Nanopore, are not really compatible with FFP samples because in many cases these samples are highly degraded, but this is not a problem to perform Hi-C. Another advantage is that you will not need to invest money in a platform or equipment like a PacBio sequencer or a BioNano optical mapping platform. We have also seen that is very sensitive or more sensitive than whole genome sequencing or RNA sequencing. In some cases, we see that 100 times more sensitive, and this is because of the nature of our chemistry and the nature of the proximity link read that you are generated with a Hi-C, meaning this Hi-C chimeric reads that are detecting interactions. And in consequence, we need less sequencing depth just to detect same genomic rearrangements in comparison to other or traditional NGS approaches. It's as I showed before, it helps to detect genomic rearrangements that are missed by other technologies. I told you in the previous slide, we have analyzed more than 400 driver negative patients samples, and we've been able to detect in at least 50% of those samples novel or gene fusion that were missed by other NGS technologies. Hi-C also provides you information about whole chromosomes involving rearrangements, meaning that with only one assay, you are going to be able to detect genomic rearrangements happening in different regions in the genome. And last but not least, the Arima Hi-C technology will also link this SBS to gene regulation by mapping de novo 3D genome structures that are happening due to the new induced by these new rearrangements that are happening in the genome. So you will ask me, okay, how can you explain that your technology is more sensitive than conventional NGS methods? So basically, because any NGS approach that you will use that is based on the use of whole genome sequencing or RNA sequencing will need to detect those reads that contain the breakpoint information just to be able to detect a genomic rearrangement, in this case, a gene fusion. When using 3D genomics, and as I explained in my previous slides, there are a lot of reads that you will generate that are informative of the interactions that are generated by this new fusion event between two genes when there is a rearrangement happening. So we have observed that when you perform Hi-C, you are obtaining even 100 times more reads that are informative of this rearrangement happening in the genome. And that's why we believe that 3D genomics helps to find those novel rearrangements that are missed by NGS, other NGS methods in driver negative patient samples that we've been analyzing. So, as you can imagine, this is very relevant for any researcher performing or doing cancer research. And then we are offering right now what we call Arima SBS detection assay. This is basically a bundle of two of our kits, the Arima Hi-C or Arima FFP Hi-C kit to perform the Hi-C reaction, our Arima library prep module to generate the Hi-C DNA libraries. And then we also bundle these two kits with some analysis or credits that we offer to analyze your data by using a cloud-based bioinformatic platform that we have also developed to detect structural variants by using different types of samples. Okay, so basically Arima offers you not only the reagents to perform the Hi-C experiments, but also a bioinformatic platform that is very simple to use and will require only that you will upload your sequencing raw data into the platform, and then you will get already a report with all the genomic rearrangements that has been detected in your Hi-C experiment. This is something that we can also provide you some information if you would like to dig into how this platform works. We have a link that we can provide you. And yeah, that's basically what we are offering for the SBS detection. And this platform, I just want to mention that has been developed in collaboration with BasePair, which is a well-known sequencing company worldwide. So we are now getting to the last part of my presentation, which I think it's going to take only a few minutes. And I would like just to give you an overview of the products and services that we provide at Arima Genomics and that you can obtain through Prodotti Gianni at Arima in Italy. So basically, we are offering what we call genome-wide Hi-C kits that will allow you to perform Hi-C genome-wide, but as with other NGS technologies, we can also offer you targeted Hi-C technologies. And then within these targeted technologies that we call capture Hi-C kits, we have a pre-designed promoter capture Hi-C kit, which allows you to generate a promoter enhancer interactome, but we can also design and produce a custom capture Hi-C kits if you are interested in interrogating the 3D genome structure of a specific region of interest. Information about our products, you can get them from Prodotti Gianni, but here I just wanted to give you a very brief overview of what we offer. Arima Hi-C kit. This is a kit that we position for gene regulation studies, DNA repair or DNA replication, and also structural variants detection in any type of samples except of FFP. Okay, this is a kit that it will allow you to generate a genome-wide map of chromatin interactions. We also have our Arima FFP Hi-C kit, which allows you to detect structural variants in FFP samples. We also offer what we call Arima high coverage Hi-C kit, which is mostly used for genome assembly of de novo sequencing sequence genomes. So normally this is just more for researchers focused on plant genomics and animal genomics, that they are really sequencing new species and they want to assemble the genomes by using a Hi-C. So something probably not relevant to you. And then we have also what we call the capture Hi-C kits, which I briefly explained you. So there are different variants of capture Hi-C kits. We can offer you the promoter capture Hi-C kit to generate this promoter enhancer interactome, a very nice solution to combine with any other omics approaches like ATAC-seq or RNA sequencing when you want to study, for example, gene regulation. But we can also design capture Hi-C kits when you are also studying specific variants or mutations in the genome and understand how these mutations could also affect a 3D genome organization in specific genes. Or we can also design a capture Hi-C kit for a broader region. And for that, you just only need to send us the coordinates of the region that you want to study, or sometimes it's not only just one region, it's, for example, 4 genes of interest, and then you will send us the genomic coordinates, and then we will just design the different probes just to capture those regions, and then you will just need to perform Hi-C just to interrogate the 3D genome structure of these regions of interest. Which are the main advantages of doing a capture Hi-C methods versus genome-wide Hi-C studies? So basically, because you are already selecting specific regions, the sequencing depth needed will be lower, so meaning that you will save money in sequencing. So this is very, very, very adaptable, the capture Hi-C kits for when you want to scale up your experiments, for example, to patient samples, and then you have a lot of samples to process. So doing a capture Hi-C experiment versus a genome-wide Hi-C experiment will help you saving money in terms of sequencing costs, and also it provides you with a slightly higher resolution for your experiments. And again, we offer this promoter capture Hi-C kit in different formats, and also we offer a custom capture Hi-C kits. And the cost will depend on the total size of the genomic regions you want to study and the number of probes that we will need to design just to capture those regions. And with that, I will just finish this presentation by also mentioning that Arima Genomics is offering Hi-C services in California, meaning that you do not only have the option to buy our Hi-C kits to perform the experiments in your lab, but you could also send us the sample, and we will also do these Hi-C experiments for you. And then the services include study design, sample prep, library prep, sequencing, and also the bioinformatic analysis. So I'll leave you here some links that you can consult if you want to look for literature, also to understand how the technology work and get more details about it. And otherwise, please contact Prodotti Gianni if you have any questions about our products.